You there? Yeah. Let's continue. So we were discussing about changes in freezing. Can you hear me? Can you see the slide? Yes. ఉన్నారమ్మా? స్లైడ్ కనిపిస్తుందా? ఆ? కనిపిస్తుందా? ఓకే. Okay, we were discussing about changes in storage. So what are the two changes that you see during storage of frozen produce? Physical changes and chemical changes. Okay. So what are the two changes?
Physical and chemical.
Yeah, physical changes and chemical changes. So under physical changes, we have seen recrystallization. So under recrystallization, the smaller ice crystals join together to form a bigger ice crystals. So as a result of fluctuations in temperature. And sublimation is the one so where the solid component directly goes into the gas component, so which leaves as a scar or a damage or a dry grainy spots on the surface of the fruit. And then and we have seen denaturation. So did you understand denaturation? Huh? So denaturation is nothing but, say, in particular cell, right? So these are the proteins which are distributed, right? So what will happen? So there is a water molecule. So these water molecules are present everywhere, distributed, right? So when these water molecules are distributed everywhere and they get dispersed, this protein gets dispersed, right? But when this particular substance will become frozen, so what will happen to the state of the food? The food will become frozen, and then so what will happen to the uh, water component? So they will become ice crystals. So water molecules will become small ice crystals. So then these proteins, the existing proteins, what are all present in the existing proteins? So they will form like a they join they join with each other to form a complexes to form. So if the protein is changing its shape or its structure, that means it is called as a denaturation of protein. Protein loses its very ability to function because of loss in its structure. Did you get that? What is denaturation? It is simple, like the proteins, so which are not frozen, they get together and form bonds with each other, and then they will form unique proteins. So that means they lose their structure, so they are not no more useful to the utilization or absorption by the body. So this causes toughness in the frozen food. Did you understand? Right. And the chemical changes during storage. So these are the chemical changes that you see during storage of foodstuff. All the chemical reactions, so they won't be stopped. They will be carried out at a slow pace, like various chemical reactions, like your lipid oxidation, Maillard reactions, enzymatic browning, deterioration of the flavor, protein insolubilization, right? This is a part of denaturation and degradation of chlorophyll, vitamins. So all these occur, these chemical reactions will occur even at a temperature of minus 18 degrees Celsius, and reduce the rate of non-enzymatic catalyzed reactions. As the temperature is lowered, so these non-enzyme catalyzed reactions will be reduced. So where the reactions which doesn't involve enzymes, so such reactions will be carried out at a lower rates. So that means they won't be stopped at a temperature of minus 2 degrees Celsius. And accumulation of off flavors. So in frozen product, you will get accumulation of off flavors and even oxidation of lipids. So that will cause again off flavors in thawing of the food, and then enzyme catalyzed oxidative browning. So this enzymes will cause browning of the foodstuff, which will reduce the appearance, the very appearance of the frozen food and your fresh food. You can easily make out because of this reason. So in a frozen food, there will be series of um, oxidative browning reactions, which will hamper, which will damage your appearance. So the food looks no more fresh. And catalysis of your ascorbic acid. You can't preserve the ascorbic acid even at low temperature. At a very low temperature, you cannot save it. Save this vitamin C. And here you can see the oozing of metacryatic liquid. So this is unfrozen concentrated solution of your salt, sugars. So this starts oozing out. So that is called as metacryatic liquid. So that means any chemical reactions, various chemical reactions, they will be continued, maybe at a slower pace, or they may be continued. They may bring about changes in the oxidation and lipid peroxidation, and the vitamin C will not be preserved. So these are the chemical changes that you see in the case of uh, frozen food. Thawing. You know what is thawing? When you keep a fridge, freezer, your meat, fish, or anything, you can't directly use it for cooking. So what you need to do? You need to thaw it. You need to bring it to your normalcy. So you need to remove that ice content in it and then bring it to the fresh state. So this can be done by thawing it. So for thawing, you need to place this foodstuff either in hot water if you have fish, or any kind of non-vegetarian food or any food. Put it in a hot water, so allow it to stay for some time, so that there will be heat transfer between the foodstuff and then the hot water, and there will be um, conversion of the frozen food to a normal food. So during this freezing process, what will initially happen? Outer structure, outer surface will be having ice crystals followed by the center portion. So this is freezing. So you have two stages. So one stage is freezing. So you have a foodstuff, and there will be formation of ice crystals on the surface of the food, on the surface, right? But we have another situation where the reverse process. This is freezing, right? The food process, during thawing process, the food passes slowly through temperature near the melting point where chemical reactions and recrystallizations occur. So this thawing occurs from the source of heat. So here it starts melting again, right? So rapid is the thawing and the recrystallization limits the time cells. So if it is not done rapidly, it will cause formation of bigger ice crystals. The bigger ice crystals will form during thawing if it is not done rapidly. So this will lead to the determinant changes like your damage to the tissues. So that will be a problem in the case of thawing. Understand? Thawing is nothing but heat transfer by conduction. So you will be transferring the heat of a frozen substance by keeping in a microwave or by keeping in hot water so that it will regain its original state. So this is thawing. And these are the methods by which of vegetables can be frozen. So here there's a beans, carrot, peas, cauliflower, guava and orange that can be frozen by using this technique of freezing process, right? So how they are frozen. So initially they need to be cooled, then packed in polythene bags, seal them, arranging them in cartons, and then freezing by plate freezing, and then storage at minus 18 degrees Celsius, right? So this is a simple process. In the case of carrot, so additionally you'll do the peeling process, peeling of the skin, cutting into 2 centimeter cubes and blanching before freezing them for 3 minutes, and then followed by cooling, and then packing in polythene bags, sealing and arranging in cartons and freezing it. So for the carrots, so for any kind of food, so blanching will help in killing the microbes, killing your enzymes. So because at freezing temperatures, you are unable to control the enzymes. The enzymatic actions continue to take place. So to prevent that one, what you need to do is blanch it. So blanching will help in denaturation of these enzymes. So when the enzymes get denatured, they won't carry out these reactions. And then blanch it and put it in the polythene bag, seal it and freeze it. It's a simple process. So for even gavas, you need to place them in lye solution for peeling. So gavas are placed in 2% lye. What is lye? 2% lye means
OH.
huh? Sodium hydroxide or potassium hydroxide,
NaOH.
NaOH. So put it in the lye. So that is an easy way of removing the skin of guava. And then wash it thoroughly. So dip it in 0.5% citric acid, cut into 4 pieces, and inside the seeds you scoop out. So you just need the mesocarp. You don't need the seeds and the mucilaginous part. You just remove it and pack it in the polythene bags. Cover it with the 25% sugar syrup. So with 100 milligrams of ascorbic acid. Seal it and arrange in cartons and freeze it. So here you are saying, so which method is being followed in the freezing process? For gavas or beans or peas, which method of freezing that you have studied? You have studied three quick freezing methods. So in that there are three methods, that is direct contact, indirect contact and air blast freezing. So for gavas, So we are placed arranging in cartons and then freezing at -1 to -5 degrees Celsius. So which method is being followed?
Indirect.
Yeah, indirect method of freezing. So where you place plates, two metallic plates, in between the metallic plates you place this cartons, and then the plates are allowed to freeze these cartons, where this will further freeze the guavas. So here in the case of your oranges, so the peeling is done, and then the segments are separated, and they are again washed with 1% lye, and again thoroughly remove that lye solution, dip in citric acid, remove the seeds. So that means here they are using segments, and packed in polythene bags and covering with syrup and ascorbic acid, sealing, arranging in cartons and freezing at -1 to -5 degrees Celsius. The process is similar for guava and oranges. So, for in the case of your fruits, in the case of vegetables, the process, the pre-treatment followed is blanching, right? And peeling is carried out in the case of your peas or fruits like your guavas or oranges. Pre-treatment with hot lye remove the peels and the pith, other aspects, and then filling it with the sugar syrup, right? So this is how freezing is carried out in the case of your fruits and vegetables. In the case of vegetables, a pre-treatment of blanching is carried out to prevent the action of enzymes. In the case of your fruits freezing, so what you do is you need to take up the pre-treatment of using hot lye solution. So that will take care of the peeling, and then filling it with sugar syrup. Here, in the case of your vegetables, you use brine. In the case of fruits, you use sugar syrup. Clear? Okay, that's all for freezing. Okay, hope you understood it. అర్థమైందా అమ్మా?
Yes, sir.
పాపం ఏం అర్థమైందో ఏమో. ఆ? కష్టం కదా ఆన్లైన్ క్లాస్లో. So that time I made a YouTube video. I was very confident that you have seen the video. I just need to brush up. So I don't know whether some of you might have got connectivity, some of you might not.